This video is on the cerebellum, its input and its output, and some of the funny names of the nuclei and components. Here is a sagittal section through the brain. The two important components of this picture are the cerebellum and the pons. The pons contains the major pathways into and out of the cerebellum. If the cerebellum is drawn out and flattened, one can see the different sections. The lateral aspect of each side is the cerebrocerebellum, or the major component of the cerebellum. It receives input from the cerebrum and is involved in fine motor movement, planning, and executing of this movement. The medial section is the spinocerebellum, and it is the only part of the cerebellum to receive input from the spine. It controls the movement in the distal muscles. The very middle of the spinocerebellum is the vermis, which controls the proximal muscles and some eye movements. The last section is the vestibulocerebellum, which is part of the most inferior area of the cerebellum and contains the nodulus and the flocculus. It is primarily concerned with posture and equilibrium. This chart shows the input connections to the cerebellum. The frontal and parietal lobes project their input to the red nucleus in the brainstem and the pontine nuclei in the pons. The red nucleus projects its input to the inferior olive. Both the inferior olive and the pontine nuclei cross the midline and send their signals to the cerebellar cortex and deep nuclei. The pontine nuclei crosses over in the middle cerebellar peduncle, and the inferior olive crosses over in the inferior cerebellar peduncle. This is, in essence, a double crossing over. The right brain controls the left side of the body, but since the right brain crosses over to the left cerebellum controls the left side of the body. The output from the cerebellum is from three areas. The first is the cerebrocerebellum. This projects to the dentate nucleus, then upward to the premotor cortex, which is concerned with motor planning. The spinocerebellum projects to the interposed and the fastigial nuclei, and then upward to the motor cortex and the brainstem. Thirdly, the vestibulocerebellum projects to the vestibular nuclei and then downward to the lower motor neurons of the spinal cord and the brainstem. The dentate nucleus, the fastigial and the interposed nuclei, and the vestibular nuclei lie deep in the cerebellum and are known as the deep cerebellar nuclei. This figure shows the functional organization of the cerebellum. The cerebral cortex that projects to the dentate and interposed nuclei pass through the superior cerebellar peduncle to the superior colliculus. The cerebellar cortex also projects to the vestigial nucleus, which passes through the inferior cerebellar peduncle to project to the superior colliculus and the reticular formation. The cerebellar cortex can project its signal directly to the vestibular nuclei of the brainstem through the inferior peduncle. The reticular formation and the vestibular nuclei send their signals to the lower motor neurons of the spinal cord. In this picture, we can see the microscopic makeup of the cerebellum. The main neuron is the Purkinje cell, the cell with the arborization of its dendrites. The dendrites of the Purkinje cell make up the molecular layer of the cerebellum, while the Purkinje soma makes up the Purkinje layer of the cerebellum. Axons from the climbing fibers synapse with the Purkinje cell dendrites. The mossy fiber axons come up to the cerebellar cortex and synapse on the granular cells in the granular layer of the cerebellum. The granular cells send their axons to the molecular layer, where they run as parallel fibers through the dendrites of the Purkinje cells, much like a telephone wire runs through a line of trees on the boulevard. This is a close-up of the interconnections within the cerebellum. The main neuron is the Purkinje cell. It receives excitatory input from the inferior olive through the climbing fibers, which wrap around the dendrites. The mossy fibers send excitatory input through the granule cells with their parallel fibers. The output of the Purkinje cell is inhibitory to the deep cerebellar nuclei, the dentate, fastigial, and interpose. This produces two loops, the deep excitatory loop and the cortical inhibitory loop. The deep excitatory loop is the excitatory input directly to the deep nuclei from the inferior olive and the mossy fibers. The cortical inhibitory loop is the projections to the Purkinje cells from the granular cells and the inferior olive, which produces an inhibitory output as part of an error correction to modify motor movement. I want to discuss prions for a bit. A prion is a protein that folds incorrectly, so it is malformed. The protein is a typical protein found in the neuron. When everything works correctly, the protein is made, it does its work, and then is broken down enzymatically and recycled. When the protein folds wrong, it can't do the work it is intended for, and it cannot fit into the active site of the enzyme, so it can't be degraded. It therefore starts to clog up the neuron with unwanted proteins, and the neuron is no longer able to function and will die. This causes motor weakness and dementia associated with Creutzfeldt-Jakob disease. It is progressive and there is no cure. When looking up the pictures online of the deep cerebellar nuclei, this picture popped up of a Majungasaurus. I had no idea. This ends the video on the cerebellum.